So for example, let's say this is a big long protein molecule. And for example, let's say that this would be the receptor site for some smell or taste. So for this receptor site to be activated where it sends, say, a signal to your brain, a smell molecule has to fit in that site. Okay, so if this squiggly line here represents the protein molecule, the folding of this is going to be the tertiary structure, but in this part here, that's going to represent the primary, secondary, or quaternary. Okay, so note that this is the shape of just a small portion of the protein molecule. So yeah, that would represent the secondary structural level of protein. So if we go back to the slide here. To answer this question, the structural level that determines its activity and function would be the secondary. So it's really what's happening in the receptors has to do with small portions of the protein which help it form. So when a protein is denatured, what does that mean? Yeah, which structure? The primary, secondary, tertiary, quaternary. Yeah, so it's usually the secondary structure. Turns out that if we go back to this slide here, we talk about structural biology, employs techniques such as X-ray crystallography. So this has actually been a very hot topic of research in the last 10 or 15 years to get an X-ray, do an X-ray experiment on a protein. Note that a protein is a big molecule. Well, you have to grow a crystal of a protein, which is very difficult to do in general. Okay, so if you can figure out a good way to grow crystals of proteins, then you can figure out the structure. Now, we had a speaker come to Hartnell several years ago, and this speaker was saying that, well, what they were able to grow these crystals of proteins using, for example, in the solvent they were using was ethanol, which is an alcohol. Now, note that in our body, the solvent is not ethanol, it's something else. What's the solvent in our body? Water. Okay, so note that ethanol is a bigger molecule than water. So my question is, well, you're claiming that the ethanol molecule, so for example, note that they actually found ethanol molecules within the protein molecules. And I said, well, since ethanol is bigger than water, you're not really showing the actual structure of the protein molecule, because in our body, the protein molecule folds in water, not in ethanol. But then this researcher was saying that, well, what they did is they put the protein back in water, and the protein worked like it was supposed to work. In other words, it went back to its native form. So I asked, well, how do you know it didn't? How do you know you didn't denature the protein? But then when you put it back in water, you renatured it again. But you're saying, well, it must have worked because when they took that crystal of protein and redissolved it in water, it worked again. So I'm always pretty doubtful about, these different solvents to recrystallize the proteins and actually whether that's the actual structure of the plant. So anyway, like I said, if you can figure out a way to grow these crystals of proteins, then you can make yourself famous by determining the structure of proteins. Now again, where do proteins come from? Amino acids? Well, proteins are long chains of amino acids. Maybe the question I should ask is, where do amino acids come from? Yes, that actually comes from RNA and nucleic acids. So, okay, so let's go and actually take a look at this. So here's an example of an alpha helix here, and here's a beta sheet here. So note that hydrogen bonds hold the those secondary structures together. So here's another alpha helix, there's another beta sheet, and in here, note that here are some, for example, this would be, for example, part of the amino acid. Okay, so I just showed, here's our protein chain, here's part of that amino acid, here's another part of the amino acid. So note that here's, it could be a hydrogen bond, which connects this part of the protein chain to that part of the protein chain. And note that in other cases, for example, note that we have two methyl groups here. What chemical force holds these two methyl groups on two different amino acids together? Because that would be an example of one force. And here's an example, for example, in our hair, we have these sulfur proteins. If you have curly hair, it's actually these sulfur-sulfur bonds which holds your hair together to make it curly. And if you want to straighten it out, you have to break these sulfur-sulfur bonds. So what chemical force holds together these two sulfur atoms here? No, not ionic. It's not even ionic. It's actually covalent. And then here's another one here. So note that in this case here, you have the NH3 plus ion, and on this other one here, you have an O minus ion from the carboxylate. So what's going to be the chemical force which holds these two parts of the protein together? Okay, so it could be a hydrogen bond, but hydrogen bonds are actually weak compared to this other force. So we have a positive charge next to a negative charge. That's a, not a covalent, like ionic bond. Okay, so in biology, these ionic bonds are often called salt bridges. So that's an example of salt. Okay, let's go on the next slide. Fats and oils are found in nature as triglycerides. Cooking oils are mixtures of fatty acids. Okay, so here's a few websites on fatty acid composition, properties of oils.
Denaturing a protein is when you change what?
Yeah, so when you denature protein, all you do is unfolding. You're changing the folding of the protein. So for example, if you completely denature this protein, you would straighten out this protein from all these folds to a straight protein.
And you use different.
Oh yes, so there's a few ways to do that. One way to denature a protein is by using an organic solvent, for example, either alcohol or gasoline or benzene or something like that. Another way to denature a protein is to heat it up. Another way would be to, okay, so you can either heat it up or you can cool it down. Another way would be to, since these are amino acids, we know that means that acid and base properties. So another way to do that would be to change its pH. Okay, so either add an acid or add a base. And another way to denature a protein would be to, so for example, it's not a good idea we're always told be careful around lead or mercury, because lead and mercury are examples of heavy metals. So heavy metals can also do the same thing. What heavy metals do, these heavy metals tend to be large ions, so the large ions can disrupt, for example, the hydrogen bonding or maybe the salt bridges in those proteins, and that's going to change the folding of that protein. Okay, so again, if it's big heavy metals, actually, they do either a good job or a bad job, changing the folding of the protein. Okay. So let's talk about lipids. I've drawn here the structure of a triglyceride. So a triglyceride consists of 3 fatty acids. So here's one fatty acid called fatty acid A. Fatty acid B is one in the middle. Fatty acid C is here. So note that the fatty acid, you have this long hydrocarbon chain and then an acid group at the end. Now note that this functional group here is not an acid functional group. It's, what functional group is here?
Ester.
Yes, an ester. Now how do you make an ester? you take a acid and react it with a alcohol. So it turns out here that, note that if we break this bond, actually it turns out that the way we do here, if we break this bond here, okay, and then connect an OH to that carbon, that makes this triglyceride into this fatty acid. And then note that this oxygen bonds to a hydrogen and becomes an alcohol. So it turns out that when we take the triglyceride, a triglyceride can undergo hydrolysis to form a fatty acid. Okay, so this will form 3 fatty acids plus glycerol. Okay, so triglyceride, has 3 ester functional groups in it, and then glycerol looks like this. Okay, so this compound has 3 alcohol functional groups in it. So glycerol, you may have seen this in the lab. It's either called glycerol, it's also called glycerin. And some of you may have that at home. If the glycerin is this sort of thick, viscous liquid, sometimes used as what? As a lubricant. Oh. Okay, so again we have this triglyceride, there's 3 ester functional groups, there's 3 chains. Fatty acid A, actually we'll call it chain A, B, and C. So in question 1A, locate the saturated fatty acid chain. Is it going to be A or B or C? So I'd like you to think about that for a minute. Once you come up with an answer, share your answer with the person next to you. Do you get the same answer?